Hello and welcome back. So today we're going to be discussing the kingdom Protista, or the eukaryotic protists, which is chapter 28 in your book, which is quite an interesting bunch. In the last lecture at the end, I mentioned that this kingdom is quite like science's junk drawer, and I'm sure we all have one at home. We have a drawer somewhere, that is filled with things that just really don't go anywhere else. It's where you've got your batteries, scissors, string, tape. I think we all have one. It's just that weirdo drawer that we all look in and go, is this a thing? And again, yeah, we have one in biology as well. There are some creatures that just do not really belong anywhere. They're eukaryotic, so they're not in archaea or bacteria whatsoever. They're single-celled. Some of them live in a colony. But on the most part, these guys, they're not fungus, they're not animals, and they're not plants. But all of them will exhibit traits of each one of those, which is really interesting. So these guys are, that's why we often say, and it's not just me, a lot of people pretty much consider the protists to be the junk drawer of biology, in the fact that they're kind of like the other eukaryotes, but not at all. So it's an interesting bunch. Again, I'm sure you've seen this video before, but I'm not going to show it here or else it will, flag me for stealing the video. But long story short, again, it's good to kind of understand the comparative sizes between what we're talking about again. So we're jumping from when we just talked about prokaryotic cells, which is the, archaea and bacteria. Now we're getting into the eukaryotic cells. So these are the big boys. They have, oops. Boop. Yeah, we can't show too much of that. But again, size difference is huge. And that's why I like showing that video, even though I show it to you probably more than once in this course. Simply because it is kind of mind-blowing to know how tiny a virus is compared to a bacteria. And then when we go from bacteria to eukaryotic cells, both protists, fungi, plant and animal cells, they're huge compared to prokaryotes. So when I say prokaryotes. So when I say, prokaryotes are small, yeah, they're not as small as viruses. They're honking huge compared to viruses. But when I say protists are small compared to eukaryotes, oh yeah, I'm talking big time. Eukaryotes are big boys. And protists actually there's one protist that's actually called, it's got a definitely piratey name. It's called the something's eyeball. And that's literally one protist, and it's about the size, bigger than your fist. And it's a protist. So these guys can get honkin' big. Honkin' big. So anyway, protists belong to kingdom Protista, which includes mostly unicellular organisms that really don't fit into the other kingdoms. A lot of them have definite throwbacks to some of the kingdoms. One in which sometimes the plant kingdom likes to claim. We'll talk more about him later. So anyway, again, they're cells with nuclei. So we have nuclei, we have a nuclear envelope, they have nuclear pores, they have organelles, so they have membrane-bound organelles like the mitochondria. Again, we saw where the mitochondria probably came out of alpha protobacteria and merged with probably a eukaryotic cell at some point and just eukaryotic said, hey, I'll protect you and give you all the sugar and oxygen you want to turn it into ATP for me, and you can hang out and be safe. And the mitochondria said, I'm down with that, and moved in. And ever since, we've had mitochondria. And then way later down the road, plant cells and chloroplasts did the same thing. Although it wasn't probably plant cells, again, it was probably, protists that could do photosynthesis by saying, hey, chloroplasts, do photosynthesis for us and we'll protect you and go sit in the sun. And the chloroplasts said, eh. So anyway, again, they have a cytoskeleton of microtubules and microfilaments, some of which we're going to be kind of looking at. Flagella and cilia. This is going to be a thing where I'm going to be showing you basically how their flagella and cilia are different from other eukaryotic cells down the road. They have chromosomes organized by histones. And if you remember back in 111, hopefully it was covered that, how histones, basically the DNA wraps around it, and how they unwind controls some gene expression. Actually, a lot more than we used to think. Then mitosis, so these guys do not go through binary fission like archaea and bacteria do. They go through straight up mitosis. They do have sexual reproduction. It's not always how we'd think of it, because a lot of these guys are single-celled, so they do something a little different. More like their bacteria cousins. And they have a cell wall in some cases. So again, some of these guys have a cell wall. Some of these guys don't, because they just decided, like amoebas. They don't have cell walls because it would inhibit them moving around. So you'll see some cell walls, you'll see some not cell walls. And again, this is kind of more, their cell wall is more looking like what the plant kingdom's going to have later down the road. So whereas the cell wall in bacteria is completely different from this is where we start seeing other things start rising. And the other three kingdoms do, or did, at some point pop out of this group, given, certain timing. And again, remember, this wasn't a one-time deal. A lot of things arose and it didn't work and went back to square one. So keep in mind when I say the other kingdoms arose out of this one, yes, but, not just all of a sudden there was plants. It took a bit. So anyway, remember last week, or when I was talking about it, early Earth didn't have molecular oxygen. So there was no oxygen, as I mentioned, if we get into Miss Royal's magic school bus, which would be purple. And we go back in time. If we got out of the school bus and we didn't have adequate protection, we'd be dead because there wasn't any oxygen. If it wasn't for our friends the bacteria, we wouldn't have any. So it wasn't until basically one of the bacteria figured out, hey, let's, taste the sun, if you watch that video, and use photosynthesis. Now, a major byproduct of photosynthesis was molecular oxygen, and that's basically where a lot of our atmosphere came from. There was an atmosphere then, but it wasn't one conducive to us breathing in it, except basically between the algae and primitive plants, they started pumping out oxygen. And because of this, it actually screwed up the Earth's weather a couple of times and snowballed the Earth. We'll get into that more down the road. So, so a major byproduct of this was making oxygen, and now all of a sudden there's tons of oxygen, and all the plants all the single-celled organisms going, hey, I could use that oxygen and use that with glucose, and bam, turn it into ATP, yo. And that was the mitochondria and the chloroplasts. And like I said, the mitochondria came first. We believe they came out of those alpha protobacteria. And it happened with the bigger cells, took them in and started using all this molecular oxygen that was now all over the place. And it's called endosymbiosis. So when one cell engulfs another but doesn't eat it or break it down, but just actually makes it a part of itself, which is what the mitochondria did, which is why the mitochondria has its own DNA. And the same thing with the chloroplasts. It also has its own DNA. And remember, plant kingdom has both mitochondria and chloroplasts. So don't get that. Some people sit there and think, well, they got the chloroplasts. They don't need the mitochondria. Oh, no. They need the mitochondria to use that sugar to turn it into ATP. So their mitochondria actually snuggles right up to their chloroplasts. It's actually kind of interesting. So again, this is where they merged for protection purposes, and they were sick of working and making all this ATP and then getting eaten by a bigger cell. So instead, the bigger cell just said, hey, I'll protect you and give you oxygen and sugar to eat, and you make me ATP. And the mitochondria said, sweet. And down the road, chloroplasts did the same thing. They said, hey, mitochondria's got a sweet deal. Can we do that too with you plant type peeps? And the plant kingdom, well, the beginnings of the plant kingdom said, yeah, okay. So, dun, dun, dun. And that's where we get mitochondria and chloroplasts. So again, here's our boy talking about it, or knockoff Brad Pitt. I will never not unsee that. Anyway, so characteristics of these guys, their cell structure. These guys are weird. Again, this brings us back to, this is the junk drawer of biology. They are just weird. So some of them can be unicellular, some of them are multicellular, some of them get huge and look like slime. We get to talk about them in a minute. They were some of my favorites. They can either have animal-like cell membranes or plant-like cell walls, and they even some of them have shells that are silica-based. Those are the diatoms. If you ever want a gorgeous time looking something up, look up diatoms. They're absolutely gorgeous. It's like geometry gone beautiful. It's like little pieces of geometry glass. I love diatoms and their little shells, which is actually called by another name. We'll get there in a minute. The metabolism, these guys can be heterotrophic or autotrophic. Excuse me. Or photoautotrophs. So again, some can be mixotrophs, which means they can do either. I love the person that came up with that term, mixotrophs. So basically, some of these guys can do whatever, as long as certain nutrients and certain conditions are met, they can either be autotrophic or heterotrophic, depending on what they need to do. A lot of them are motile. Some use flagella, cilia, or pseudopods, which means fake feet. The ones with the pseudopods are like amoeba-like. Slime molds are in that group too. Slime molds make pseudopods, bleh. So imagine if you were a big blob of jelly and you could just go, yeah, that's basically what a pseudopod is. So they can move away or towards a stimulus, which is known as taxis, like phototaxis. And if we had a lab, I know this is the summer and it doesn't really show. It's like we would go out to the big pond lake thing that's on Blue Ridge's campus, and we'd be getting things. And some of them love to run right for the light. They'll come right up and stare at the light on your microscope, and some of them run off because they hate it. So it's annoying when you're trying to look at the ones that run away and you're just chasing them around under your microscope. It happens. It's happened. It will always happen. Because some of them like the light, some of them don't like the light, and some of them just don't care. So again, that's the fun part of trying to take pictures of these guys when they're running around like goobers under your microscope. Anyway, they have crazy life cycles. Most are asexual, either through binary fission or mitosis, but they do have sexual reproductions where they can switch between the two. Again, we've got some people that are like, yeah, well, I can just, make a copy of myself until I can do a full, reproductive thing. So again, depends on the environment. Some of them can do both. You're going to see a couple of life cycles. I'm going to show you a slime mold life cycle. I'm going to show you one other. They also have something called cysts. So this is like what the bacteria could do in the fact that they form a protective resting stage until the environment can support reproduction in earnest again. So they can ball up again, wait for the environment to get better, and then finally go, oh, I've got enough energy, and the environment is just right. It's time for reproduction. So, habitats, they're everywhere. There isn't an inch on this earth that isn't covered in protists in some form or fashion. We've got them everywhere. So they exist in some sort of aquatic environment. As long as there's water, they're there. Freshwater, marine, damp soil, snow. We've found them even in the snow in Antarctica. That's just how much we've seen these. Several protist species are parasites that infect animals or plants. Again, we'll have another the good and the bad slides at the end of the protists we love very much, and the protists that we wish didn't love us so much. So again, some of them live on dead organisms. Again, these guys help break things down, just like bacteria do, or their wastes, or contribute to their decay. So we do have some decomposers in this group. So it's a whole party. Now, we have, instead of phyla, we have supergroups in this one. I don't know who called that, but anyway, not me. So the 6 supergroups that we are going to talk about is Rhizaria, Chromoviolata, excuse me, ciliates and dinoflagellates in there. They're fun. Archaeoplastidia, which is green algae, red algae, and what we think the land plants popped out of. Now, green algae is in a tug of war. We'll talk about this more over in the plant kingdom a little bit, but green algae is kind of in a tug of war between Protista and the plant kingdom. Depending on the scientist and depending on who's doing the classification, green algae can almost go either way, because green algae pretty much gave arose from which arose the land plants. So these guys basically gave rise to these guys. So that's why they're all hanging out in that group. Amoebozoa. So this is where our buddies, the amoebas, are, and the slime molds. We love them so. And I'll talk about why they're just so crazy neat. So amoebozoa is an interesting pile. They're the ones with the pseudopods. Opis theconta. This is basically a lot of animal likes and a lot of fungal likes hang out here. So we believe that both the fungi kingdom and the animal kingdom, actually I shouldn't say we believe, we have a lot of evidence to support that an animal kingdom and the fungal kingdom popped out of this group as well. So you can already see out of 2 supergroups, 2, the rest of the 3 kingdoms that we're going to be talking about came forth. So Opsia theconta has been kind of busy. Anyway, excavata has the Trigomonas, Giardia, the euglenoids, euglenas, which are funny because they look like feet and they roll around. Anyway, so these are the 6 supergroups. Again, I'm probably not going to break them down more than that. We're just going to go over them and why they're crazy fun. So again, Archaeoplastidia. So molecular evidence supports that pretty much all these guys are descendants of the endosymbiotic relationship between cyanobacterium and a protist. Again, which probably became the chloroplast. So this group includes your red algae and your green algae. Green algae, we like. Red algae, we've been learning how to use them for our own interesting moments. Red algae has been started growing commercially. We actually harvest them and turn them into vitamin pills, interestingly enough. There's other things we do with them too. But and if you've ever poked green algae with a stick in a pond, you know exactly what green algae is. So it was a common ancestor of these protists. The land plants came out of this group. So it's their closest relatives found in this group, which is why sometimes the green algae gets somewhat... He double dips in this group and kingdom plant, depending on who's doing the classification that day. So red algae, green algae include unicellular, multicellular, and colonial forms. Like I said, most of the time, we like these guys. Amoebozoa is where the amoeba and the slime molds are. They are both weird. And I do mean that because I'm going to get into slime molds in a minute because I just think they're just the bee's knees. They're extensions, so they have the pseudopodia, the false feet, the extension of the cytoplasm. They reproduce via binary fission. So some of these guys have protective tests. Again, a test is just a protist's shell. We just have a different term for it. So if it was something else, it would be a shell in another kingdom. But for protists, they're called tests. So some of them are just don't have a shell or a test, and some of them do. Slime molds act more like fungi, and a lot of people get them confused. You've probably seen a slime mold. Sometimes they look like a dog barfed in your garden, and they're not going to hurt anything. We get rid of them because they look nasty, but they're actually doing a good job in your garden. So if you see a big yellow, it looks like, some weird fungal infection going on all of a sudden, a big yellow, don't worry about it. You don't even have to do anything to it. It'll go away on its own. It's a slime mold. You're just made friends with a slime mold, and he's your garden's friend. I know he doesn't look delicious or anything like that. He doesn't look at all fabulous, but don't hurt them because they're actually helping your garden be healthy. So, but they have a very distinct life cycle, so we're going to get into that. So again, here's a beautiful amoeba. Isn't he cute? We don't like amoebas. We like amoebas to look at. We don't like them in our water. That's how we get amoebic dysentery, and it's really bad for you. So please do not always drink bottled water when you're outside of, visiting other countries. Because you can come home with amoebic dysentery, and it's not a fun time. But we like them under a microscope. We do not like them inside of us. So again, here's, you can see he's sticking his cytoplasm out. So he doesn't have a cell wall. And therefore, he can make these little legs, which are the pseudopods. And depending on where the food is, they just literally shift all their cytoplasm that way. So anyway, now, slime molds are really interesting because they're actually intelligent. I know it kind of blows our minds. We actually, with using food, you can make a slime mold run a maze, and it will find, it will literally find the fastest route out, using pseudopods. And it's insane just to watch these guys. It just looks like, again, if you've seen this in your garden, don't destroy it. That's your friend, the slime mold. He's a protist, and he's your friend. And they do good things. So they do a lot of good things. But they have a pretty interesting life. The reason they're in the amoeboid family is because their babies, their spores after they hatch, turn into one part has flagella, double flagella, and another one has little amoeboids. And they get together and turn into a zygote. And then what you see when you see it blow up in your garden sometimes, or on, especially on, if you put down some mulch and then you go in, and then a couple weeks later you'll see sometimes this blows up on your mulch. That's the feeding plasmodium. So that's right after the zygote fertilizes between the amoeboid and the flagellated. And they come together, they form a zygote, and it blows up like that. It matures, they send up little stalks, and then when the stalks, they kind of go away, and that releases the spores, and around it goes. And like I said, they're actually really fun. You can make them run a maze by using food. We've actually also used them to plan out cities. I'm not kidding you, because they take always the most expedient route. There's some Japanese scientists who were testing on them. They decided to give them the lay of the land using food and things that they hated, where food was like the cities, and things that they hated were like water they couldn't cross, like a river, and some other landmarks you couldn't just cross. And literally, these guys mapped out the most efficient routes between the cities and all the places they couldn't go. It was... And if you overlaid it on a human civilization, on human city, and what they decided, it actually came out insanely close. Actually, sometimes even more, to how to build roads and everything, depending on where they go. So, yeah, you could use these guys to plan a city if you want. They'll figure out the most expedient routes. Pretty crazy. Maybe they should be in planning for roads and stuff. So it's crazy. Speaking of, here's a video that goes over it. It's called True Facts. If you've ever seen any of Z Frank's work, they're absolutely hysterical. Highly recommend. They're all very educational. He does get his stuff fact checked before he makes these, but they're absolute hysterical things. So feel free to watch, but watch out again. Swear words in this. Highly recommend to watch, though. Very funny. So, just FYI, if you don't want to watch the swear words, cool. But you're missing out on a really funny and really educational video. He has a lot of them. I love them to pieces. They're hysterical. If you've ever seen the one with mantis shrimp, that's him. So, good stuff.
This episode sponsored...
Anyway, Apsithecota. So this is named after the single posterior flagellum seen in flagellated cells of this group. So they had, instead of that double one that you saw over in the amoeboids, these guys have the one. So these organisms, again, the single flagellum is surrounded by contractile collar composed of microvilli. That collar is used to filter and collect bacteria for ingestion by the protist. You're gonna see this again. And the tip-off that the animal kingdom and the fungi kingdom came out of these guys, again, here comes the hiccups, is the fact that this collar with the flagella in it, you're going to see it again in sponges. Dun dun dun. SpongeBob SquarePants. Yes, exactly. That's how they filter feed. So you're going to see that collar with the flagella again when we talk about the first creature in the animal kingdom, which are the sponges. So there is a strong, lots of evidence pointing that this is where the sponges grew out of this group. And from the sponges, you know, not just the sponges, but a lot of other animals started popping out there. Because we see this collar with the flagella again and again in a lot of the invertebrates. So that's why we have a lot of evidence to suggest that the animal kingdom popped out of this group. We also think fungi popped out of this group as well. So it wasn't just the animals that popped out. And there are, interestingly enough, I know you look at a mushroom and you go, that doesn't look anything like an animal. You're right. They're not. That's why they got their own kingdom, which I'll talk about in the next lecture. So just FYI, these guys are really interesting because they went in a whole bunch of different directions, definitely. But that the collar cells, you're going to see them again. So they're mostly parasitic. Which, again, if you're parasitic, it means you're heterotroph. And again, what's animal kingdom and the fungi kingdom? They're heterotrophic. Rhizaria. So this one basically looks like amoebas with a whole bunch of like needle-like root pseudopodia. They basically have, again, these guys are really important in carbon and nitrogen cycles. So it isn't just the bacteria. These guys pop in too. When they die, their shells or tests sink deep into the water and basically lock away a ton of carbon dioxide. So they suck up carbon dioxide and take it with them and then sink down. So these guys basically make up calcium carbonate or silicates. So if we need, you know, calcium carbonate, if you've, you know, ever taken, you know, earth science back in high school or whatnot, and I mentioned calcium carbonate being one of the minerals that comes with living creatures, it's one of the few that we consider a mineral that comes from a living thing, because one of the rules that's kind of on is it a mineral or is it not is usually it's inorganic, with the exception of calcium carbonate. Calcium carbonate comes from these guys dying. So these guys with their tests or shells, when they die, they're actually locking away a ton of carbon dioxide. So we have them to thank for, you know, trapping and storing carbon dioxide at the bottom of the waters. In Davy Jones' locker. All right, Chromoviolata. So this basically is a big group that have flagella, have bioluminescence. So some of them glow in the dark, some of them are parasitic like Plasmodium, who has a very complicated life cycle. I'm going to kind of show it to you and then go, yeah. I'm not going to ask you to memorize that because that's a little crazy. Plankton, which is why I have plankton right here. Parameciums, diatoms, again, my favorite. Their tests are absolutely gorgeous because they're made of silica. I'll show you a picture in a minute. They got red algae, brown algae, golden algae, and water molds. Again, some of these algae we like to use. Some of these algae, if we get a bloom of them, it is bad. Red algae blooms can like suck out the oxygen of an entire lake or a part of an ocean and kill everything living in that section of the ocean or lake. So if you ever hear of an algal bloom in a lake, that's a bad thing because that means the algae is going bonkers and growing like crazy unchecked, and it's sucking up all the oxygen out of the water to do it, which kills off all the fish and everybody else living in there. So that's why algal blooms are not your friend unless you're growing them in a tank for some reason. Anyway, excavata. So these are asymmetrical single-celled organisms with a feeding groove. Again, these guys are interesting. A lot of them are heterotrophic predators. Some of them are photosynthetic and parasites. The euglena is in this group, and the euglena looks like a foot. And then you've got, you know, Giardia lamblia, which is a parasite, then Trichomonas, which causes Trichomonas, which is a sexually transmitted disease in humans. So the bacteria didn't have all the monopoly on the STDs. Protists got some too. Anyway, so let's go ahead and look at here's the diatoms. Aren't they just absolutely gorgeous? That's all silica. That's all their tests, which is silica. Glass, basically. Here's our buddy, the Euglene. And they just twirl when they swim around. But they have a mouth pore. They have an anal pore. Yep. They decided to have a mouth where food goes in. And an anal pore where the food goes out. Or what's left goes out. Have a contractile vacuole. Again, you'll see contractile vacuoles in a lot of protists because they need to control how much water is coming in and out of them at any given time. So they have to be able, remember, diffusion with water, osmosis, and how water can go in and make a cell pop. A lot of the protists have a contractile vacuole to push water out if they get too much water in them, and vice versa, lets them in. So it's kind of interesting. They have covered in cilia. They have food vacuoles. Anyway, water molds, these are interesting. These are the mildews. Fish tank fuzz, if you've got fish and you've ever had to fight the fish tank fuzz, it's these guys. They're interesting because they have a cell wall. So again, they kind of have some plant-like characteristics, but they're not, again, in the plant section. So, Omeomycota, interesting bunch. And again, here's our buddy with the contractile vacuole right here. He's got one too, again. He's got the flagellum. Now, the interesting thing is, for these guys right here, their flagella is in the front. So they use it as a front way instead of in the back. So they use this to flip back and forth to go forwards, not put the flagella in the back to go that way, like, you know, human sperm would or a tadpole or something. So their flagella is in the front going back and forth, back and forth, back and forth, and that drives them forward. So, you know, whatever works for them. But again, these guys like hitting the, oh wait, excuse me. Paramecium. Euglena. Yeah. Yeah. Anyway. Now, so here is a typical protist that causes, so the plasmodium, that basically, you know, causes sickness, malaria in humans. So, yeah. This is, again, I'm not going to ask you to memorize this. I just want you to realize how crazy this is. So mosquito that has these protists in their saliva, I'm not kidding about that, takes a bite out of a human, then it goes into the liver and starts infecting the liver and starts growing in the liver, then it goes into the blood, then it hits maturity, and then another mosquito picks it up from sucking blood out of a human, and then again it goes into the mosquito and forms and goes through mitosis, and again and around and around and around and around it goes. So this is what causes malaria in humans, and the mosquitoes just bring it along for the ride and infect their next victim. But this is just how crazy a life cycle Plasmodium is for causing malaria. I mean, this is nuts. So, again, so why do we like these guys? Oh, we like them very much. They are extremely important to our ecosystems. They produce oxygen. Again, these guys in bacteria produce a ton of oxygen. They function as producers in both the freshwater and the saltwater ecosystems. So a lot of creatures eat these guys. Like, for instance, plankton, our dear old friend plankton back here. Yeah, a lot of ocean biomes pretty much run on everybody eating plankton. There is a ton of plankton, and it serves as a ton of food for heterotrophic protists and animals. I mean, you know, whales eat plankton, so it's crazy. So, you know, they're suspended in the water. Many protists are symbiotes. A lot of them are strict parasites, but some of them are mutualism. Coral reefs are greatly aided by the symbiotic phototrophic in the tissues of corals. So these guys are extremely important. Again, oh, he's looking at diatoms right here. This is a Volvox, and you can actually find these guys in the waters around here. They just look like a big bubble made of bubbles. They're actually they're really cool. They're photosynthetic. You can find them around here on pond water and stuff. Here's some diatom shell or tests. They're so pretty. They're so pretty. So again, yeah, there's a few protists we don't enjoy, but for the most part, without protists, A, the next 3 kingdoms we're going to talk about wouldn't be here. And B, they do a ton of stuff for us. So thank you, Protists. Thank you, Protists, very much. And even Plankton. Because without him, a lot of ocean life would be starving. So, I believe that's everything for this. So with that said, that's chapter 28 in a nutshell. Now we're going to skip forward and kind of skip over in your book. The next chapter, next couple of chapters deals with the plant kingdom. We're going to skip that for now and go straight to the fungal kingdom, and then we'll double back to the plant kingdom. I just think it's easier to talk about the fungi first and then talk about the plants. But that's just me. So maybe it's just because I'm crazy like that. But anyway, so there you go. I'll see you in the next lecture. Thank you so much, and have a good one.